Thank you for joining us for another episode of the Diabetes Bio Podcast, a podcast for the Diabetes Journal, the flagship research publication of the American Diabetes Association. My name is Darleen Sandoval, and I'm joined by my podcast co-host, Dr. Kirk Habiger and Dr. Kevin Williams. Hello. How are y'all doing today?
Morning, Dar. Kevin?
Morning. Doing well. How are y'all doing?
Good. So we're recording this on a Monday. How was the weekend? What did y'all get up to? I'm sure there was some athletic event to go to, Kirk.
No, we're done. We're done.
Wow.
Although we are in banquet season, which is arguably worse. So don't tell my kids I said that.
They don't listen.
Yeah, but at least the sporting events are now to close. So we did actually, this weekend was the Kentucky Derby.
Yes.
And so we went to a friend's house for a crawfish boil.
Oh, fun.
Oh, nice.
And watched the horses, which I know nothing about, but it was fun.
Did you make a bet?
I did not, but I think we were at the right place where someone who was interested, but it wasn't their entire life. Right. And
Yeah.
so they were excited. So everybody was excited. But not so much so that we had to know the details of what was going on.
Yeah.
What about you guys? What'd you do?
So I just got back from being out of town all week on Friday night. So we went for a, quote, family hike, which is me, Damon, and the dog.
Counts.
And that was really nice. And then we went to have dinner. And then so usually on this weekend, the first weekend of May, I'm usually in Cincinnati to do something for the Flying Pig Marathon. Lately, it's been the half, and celebrate with friends. We had a really, really good friend that passed away some years ago. And so it's our reunion weekend. We get together, have a lunch or whatever, and cheer each other on in the race. And then usually I watch the Derby with Linda and Susan. So we were texting each other during the Derby.
There you go.
While we were sitting there having a lime leaf margarita.
So did you place a bet? Did you win any money?
I didn't, because I was a little late, and I would have bet on So Happy, so I wouldn't have won any money. But
There was a video of So Happy after I think the trainer and somebody was over there petting So Happy and did not look so happy after the race. But I didn't watch it. I've just seen a bunch of clips about it. And I did typically watch that over the years, but it's been pretty inspirational with what was the winner? Golden Tempo coming from the back on the last corner in the stretch.
That was
so
amazing.
It was amazing to watch. I mean, it's just unfathomable to think of catching up to the front of that pack in just such a short distance and the amount of effort that goes into that. And so it was really cool to watch that.
Yeah, definitely. So Damon did his residency in Louisville, so we never got to see it or anything. But although I did do the Louisville Marathon one year, and that race actually goes, I don't know if it still does, but back then it went sort of under the track into the infield, and you do a loop in the infield and then go out again. And it's usually the week before the Derby. And so they bring the horses out because it's, thousands of runners.
Mm-hmm.
And I guess what I heard was that it's a way to get the horses used to having all of the people and have them out like kind of running around the track. So it was super cool. Like if it's rainy or muddy, you don't get to see them. But the day I did it, it was really nice. And so, we got to see the horses out there running. It was super cool.
I've never been to the Kentucky Derby, but I've been to Churchill Downs, and it always amazes me that this facility is like in the middle of a neighborhood.
Yeah.
And
is that right?
Yeah. And we ended up parking like behind people's houses in grass, and it was just our random Friday. But yeah, it's an interesting, cool old place.
Yeah. Yeah. That's
sounds a lot like Wrigley Field in Chicago, just nestled in a neighborhood.
Yeah. Very cool. All right. Well, I guess we should get to work. So, Kirk, tell us about this month's articles.
Yeah, so this month we have another set of great articles. We'll start with May's paper of the month. Tomosyn-2 regulates postnatal beta cell expansion and insulin secretion to maintain glucose homeostasis. In that discussion, we were joined by a corresponding author, Dr. Sushant Bhatnagar, who just happens to occupy the lab across the hall from mine here at UAB. So that was a fun discussion, and he was gracious enough to take some questions he wasn't expecting. Following that, we discussed another article, the normalization of insulin resistance rather than hyperglycemia before stroke improves functional outcome in a mouse model of type 2 diabetes. That article was from Dr. Vladimer Darsalia from the Department of Clinical Science and Education at the Karolinska Institute, and was another great one.
All right. Very cool. Kevin, what do we have going on for Sweet Talk?
Yeah, I think we have a special one today. We're talking with this year's recipient of the American Diabetes Association's Outstanding Scientific Achievement Award. It's Dr. Timo Mueller. Timo directs the Institute for Diabetes and Obesity at Helmholtz in Munich. His lab helped crack open the brain GIP receptor story, which is foundational biology that reshapes how we understand why drugs like tirzepatide work as well as they do. We'll get into the science, where the field is heading, and what it took to get there. So I think it's going to be really cool conversation. Stick around and find out more.
All right, let's jump in.
Today, we will be discussing an article from May. It's the paper of the month titled Tomosyn-2 Regulates Postnatal Beta Cell Expansion and Insulin Secretion to Maintain Glucose Homeostasis. We are joined by corresponding author, Dr. Sushant Bhatnagar, who is an associate professor in the Division of Endocrinology, Diabetes and Metabolism in the Department of Medicine at the University of Alabama at Birmingham. For those of you wondering, he's about 4 doors down the hall from me. So this is a fun podcast. Sushant, welcome, and thank you for sharing your work with us today.
Thank you for having me. It's a pleasure.
Sushant, we like to start with an icebreaker question. And for you, I'm going to ask you, if you could acquire any new skill in the world, what would you choose?
Any new skill in the world? Music. So it doesn't have to be a science question, right?
No, absolutely not.
Yeah, I always wanted to learn to play an instrument. And
any specific instrument?
Keyboard.
Yeah.
And that prompted me to get my son into, learning keyboard. I don't know how much he's enjoying it, but I would love to get into that if I can get some time.
Very cool.
Being in science.
Yeah. Sushant, before we start talking about the science, I want to give you an opportunity to talk about any other authors or collaborators who added to this work.
Yeah, absolutely. This is really the team effort. So the first authors are Catherine and Justin Alexander, and did a huge amount of work. I mean, incredibly, they're very strong experimentally. And Haifa Al-Sharif was a very key to this project. She was a graduate student in my lab and now a postdoc at Harvard, and she really helped bring this story together. And then this paper also has a bioinformatics component. So Dr. Nguyen, he's an assistant professor at UAB, did a lot of bioinformatics work. And we had good collaborations, actually great collaborations with the labs of Jeonga Kim and Chad Hunter. So definitely a group effort, I would say.
Great. Okay, now, can you start by giving us sort of an overview of your research program and then how this specific project grew out of that?
So my lab is broadly interested in understanding how pancreatic beta cells regulate insulin secretion, maintain functional identity, and adapt to pathophysiology and metabolic stress, and also in the pathophysiology, broadly in the context of obesity and diabetes. A major theme of our work is that beta cells constantly balance competing demands. On one hand, they have to secrete insulin efficiently to maintain glucose homeostasis. And on the other hand, they need to preserve the mass for their long-term fitness. So this project fits very well within this broader research program because it addresses a fundamental question in beta cell biology. How do beta cells transition from an immature proliferative state early in life to a mature, highly functional insulin secreting state? In this study, we identified Tomosyn-2 as an important regulator of that transition. And what makes the story especially interesting is that Tomosyn-2 does not simply regulate insulin secretion in isolation. It appears to coordinate the balance between insulin secretory function, maturation, and proliferation. So this work really expands on our thinking from exocytosis alone to a broader question of how beta cell function and mass are linked in this postnatal period.
That's great, Susan. Tell us a little bit more about Thomasin-2. I believe you describe it as a molecular brake. Can you tell us about that?
So I'll take a step back and just give a little bit of beta cell and insulin secretion context. So like beta cells are designed to regulate insulin secretion very tightly, and we know that. And these cells store a substantial amount of insulin. And if you think it loosely, like if a large amount of insulin is released at once, it could drive blood glucose to a dangerously low levels, leading to hypoglycemic state. This is extreme. So in other words, both too little insulin and too much regulated insulin secretion can be harmful. And maintaining this tight balance is absolutely critical for long-term beta cell health. Now, if we look at the literature on insulin secretion sort of broadly, we know a great deal about factors that stimulate insulin secretion, but much less, very little is known about the mechanism that restrain or inhibit it. And that's where Thomasin comes in. And Tomosyn-2 is a syntaxin binding protein that negatively regulates insulin secretion. And in beta cell, it acts by interfering with the assembly of snare complexes, and which is a core machinery required for insulin granules to fuse with the plasma membrane and release insulin. So functionally, Tomosyn-2 is an inhibitor of insulin granule exocytosis. So when we describe Tomosyn-2 as a molecular brake, we mean that it places a constraint on the secretory machinery, which is driven by snare complexes. So it prevents beta cells from becoming overly active in releasing too much insulin. So that's sort of the base definition. Now, I'm glad you really asked this, because what particularly become interesting in this work is that this brake appears to serve a broader developmental role, especially in this postnatal stage. It is not simply dampening the secretion for the sake of reducing secretion, but what it does is it may help maintain beta cells in a state that allows it to expand early in life. So then Thomasin, and as Tomosyn-2 expression declines, beta cells are able to acquire a more mature state with enhanced insulin secretion. So the molecular brake concepts sort of operates in two levels. It restrains insulin secretion acutely, and then it helps coordinating the timing of beta cell maturation versus expansion during this postnatal developmental period.
I think that was a great summary and breakdown of the story. So you used a genetic model to try to understand this role of Thomasin-2. Tell us a little bit about that mouse model.
So we used a Tomosyn-2 loss in function mouse model. And the idea was to define, the goal was to define the in vivo role of this protein in beta cells. And what this mouse model did is allow us to examine how removing Tomosyn-2 affects insulin secretion, beta cell proliferation, beta cell mass, and overall glucose homeostasis across neonatal and into adulthood. Now, what we found strikingly was that loss of Tomosyn-2 enhanced insulin secretion. It's an inhibitor, so it increased its loss will enhanced insulin secretion, improved biphasic insulin release from islets, and that translated into better glucose clearance determined by glucose tolerance test. Now, phenotypically, these mice actually look metabolically better. They control glucose more efficiently. And mechanistically, this fits with the insulin, with Tomosyn-2 acting as a brake on snare-mediated exocytosis. So when we remove a brake, the beta cells became more functionally active and behave more like mature insulin secreting cells. But the key insight that we gain from this model is that gain in function of insulin secretion or better glucose homeostasis comes as a cost. And we observed that reduced beta cell proliferation, impaired expansion of beta cell mass. In other words, when cells became highly functional, they lose their ability to expand in number during this postnatal period. So although beta cells were functionally stronger, the overall expansion of mass was limited. And that combination was one of the most important clues that Tomosyn is coordinating this balance between beta cell function and growth. So this mouse model helps us sort of establish this, that there is a molecular link between function and growth. The other thing I wanted to bring out is that these effects occurred without changes in body weight or insulin action. Now, insulin action we determine by insulin tolerance test, but what that indicates is that the effects that we see in the beta cell are not driven by systemic metabolic stress or insulin resistance. So instead, it reflects the cell intrinsic regulatory mechanism. So overall, the model revealed that Tomosyn-2 functions as a molecular switch that coordinates the balance between beta cell function and expansion.
You've very well described how Tomosyn is sort of implicated in this switch. Can you maybe take us a little bit deeper into the details of those mechanisms? How do you think that it's contributing in the cell's ability to proliferate versus its ability to secrete?
This is sort of very fundamental, right? Like the beta cell biology, sort of the immature beta cells are proliferative, but they're not yet optimized for glucose stimulated insulin secretion. And as beta cell matures, several things happens. They increase expression of genes involved in glucose sensing, mitochondrial metabolism, insulin processing, secretory granules, and stimulus coupled secretion is up. At the same time, there is a reduction of genes involved in proliferation, cell cycle activity, and immature dip. So this transition involves both gain of function and loss of replicative flexibility. And mature beta cells eventually become good at sensing glucose and mounting a robust secretory response. So what we saw in Tomosyn-2 loss, and Tomosyn works at a plasma membrane level, that essentially all of the program, it functionally not only loss of Tomosyn not only functionally block proliferation, but essentially the gene expression profile were also similarly affected. Like the genes that are involved in beta cell maturation, secretory process, mitochondrial function, vesicle maturation, trafficking, all were upregulated. And then conversely, cell cycle genes, genes that are involved in cell cycle, all of those were downregulated. So it wasn't a functional connection, but also the whole reprogramming at the gene expression level that also happened. But the way we linked it back was we found surprisingly that the AKT signaling pathway was downregulated. And we know from a lot of the labs that have worked and showed that AKT signaling, when activated, increases cell cycle progression. So somehow loss of Tomosyn-2 and the protein that works on the exocytosis, insulin exocytosis, is able to downregulate AKT signaling pathway. And so we think that's the molecular link. But then somehow the whole gene expression reprogramming is also occurring. That is very striking to us.
I guess I want to ask a follow-up here. If Tomosyn-2 deficient mice have this accelerated maturity, right, and a lack of proliferation, do you expect that there'd be a deficit, or have you looked yet for deficits during times when the beta cell mass must expand? So high fat feeding or pregnancy.
So we have, I mean, that's a great question. And I think that's something, we're going to follow up with that, because right now we focused on postnatal period. And, in this postnatal, you have this sort of inverse relationship secretion over time goes up, and then the proliferation goes down. And then one key thing is adult beta cells do not proliferate. But then the key is, does this thing happen in high-fat diet? Because that's a different sort of proliferation and secretion dynamics. In high-fat diet, beta cells again starts to proliferate, but now they're proliferating to sort of compensate against insulin resistance. So is it same or not? We don't know. But these are really good questions, and that's where we are headed. That's the exactly would be hopefully our next paper.
You're answering the future direction question already.
Yeah.
Tell us, you mentioned the transcriptomic and network analysis. Some of I guess I should say, you mentioned some of the gene expression changes that you saw. Can you talk a little bit more about those hub regulators that you identified that were important in the secretory network?
Right. So, like I mentioned earlier, it was what we found is it was not just one or two genes that were affected upon loss of Thomasin. Pretty much the network genes, so loss of Thomasin, what it did was it led to increase in genes involved in insulin secretion, maturation, and identity, and also mitochondrial metabolism and exocytosis. And we ended up the network that was formed from those differentially expressed genes that were upregulated in Thomasin. We found several key known hub regulators. Insulin 1 and 2 were themselves. AKT was very central. It was sort of one of the main connecting nodes, and that's what led us to chase after AKT signaling. But then GLP-1 receptor, was there in every sort of known key transcription factors like NKX6.1, PDX1. They were also part of that network, and they were hub genes. So a lot of the known factors in the field that are involved in insulin exocytosis, maturation, and beta cell identity were there as regulators.
Yeah, interesting.
So Sushant, I'm going to dig again here, and we've discussed this project more than once. I think it may have been on my whiteboard for a while. It has always struck me as amazing that removing this protein that is usually a resident of the plasma membrane has such profound effects on the transcriptional regulation of the beta cell. And one thing that you and I have discussed is that this was a whole body knockout, right?
Right.
So any insights that you can provide here on whether this is a direct effect of beta cell Tomosyn-2 or lack thereof, as opposed to some secondary effects of Tomosyn-2 deficiency elsewhere?
Right. That's sort of a limitation of this, this project, and also, but historically we have shown Tomosyn-2 is very active. Some of the work we did that like 10 years ago, that was, we identified Tomosyn-2 mutation in Tomosyn-2 to have an effect on beta cell function, and now we are characterizing a physiological role. So Tomosyn-2, what we have shown is directly binds in beta cell. And the studies, to answer, more accurately this question about whether it's a direct versus indirect. So I think the case can be made very much in favor that it has a direct role, because what we showed in cell lines too, that we are able to get the same in the beta cell lines. Specifically, we are able, the outcome is sort of consistent between the role of Thomasin on exocytosis. So when Thomasin binds to syntaxins, both in mouse islets, we showed it proximity ligation assay, and then in mouse beta cells. And also when we do it in the beta cell line, we are able to show that Tomosyn-2 binds syntaxin, and there is more of Thomasin. We have less of snare complex formation. So Tomosyn-2 is very much active in regulating exocytosis in beta cell. So functionally, Tomosyn-2 is working in a beta cell manner. Now, can we envision that, Tomosyn-2, since it's affecting exocytosis, there could be a potential secreted protein that could be co-secreted with insulin and can sort of feedback in an autocrine or can have a paracrine effect on other islet cell types. And then there can be sort of like, some sort of, fine tuning effect. That's there. That's possibility is there. And I think that's why, we will continue working on this.
Yeah. Yeah. Thanks.
More questions is a good problem to have.
Right.
So how does this finding specifically challenge our assumptions about beta cell proliferation and maturity or the maturation process?
The field is wide, right? Like we have people have been working on proliferation, or investigators for the last several decades on secretion and proliferation. And I think this connection is more recent, where there were papers where they show that there is inverse relationship between these two. Traditionally, yes, we often think that the beta cell proliferation and beta cell function are sort of these desirable attributes of beta cell that could in principle be increased together. And this is, I'm talking about broadly, right? Like if someone is really in tune, there. But broadly speaking, I think it's like, we can both increase proliferation and function. But what our findings are highlighting is that they are sort of inversely linked, at least in this postnatal developmental context. Now, high-fat diet is to be determined, right? Pathophysiological state is to be determined. But at least in this study, that enhanced function and maturation did not coincide with the greater proliferation, and it in fact came at an expense of expansion. So basically, this work suggests that beta cell may face some sort of a biological trade-off, like the same process that supports mature glucose responsive insulin secretion will limit proliferating capacity. That changes how we think about sort of therapeutic strategy. Now, this is an extrapolation, right? Like, because this study is focused on neonatal, but one can imagine that if the same thing happens in high-fat diet, then what that means is just that's simply pushing beta cells to become more functional may not necessarily preserve or expand beta cell mass. So maybe the strategy could be to find ways to uncouple these two processes in some way. So maybe, you have drugs that target proliferation and then and secretion independently. But maybe if what we are seeing in neonatal happens in response to high-fat diet situation, then pushing one could be maybe not ideal.
Yeah. It's also interesting to think about if mom is obese, right, or has obesity, then what happens to this process?
Right. And I think there is some precedent to it. And I was talking to one of our clinicians, and they mentioned that sometimes in a segment of population, what they have seen as I would say kids, maybe the children's would have hyperinsulinemia. They have more, their pancreas have more insulin, and if in some sort of, and those are the ones when they were followed up till the adulthood. I think there are studies that link that those are the ones that have more susceptibility to developing type 2 diabetes. Now, I'm not that familiar with the literature on that one. I would need to read. But what you're saying is, maybe, I think it's again important to study.
Definitely.
Well, Sushant, I think we've had a pretty good discussion on Tomosyn-2, its role in regulating the opposing proliferation versus secretion biology of the beta cell, and some places where you want to take this project. But I do want to give you a chance to give us, maybe one key takeaway or maybe another future idea before we leave.
So I think the one major takeaway is that beta cell maturation is not simply the acquisition of better secretory function. It is also accompanied by the loss of proliferative potential. And it is this balance that is very essential, at least in this neonatal period. And Tomosyn-2 appears to be important regulator of this balance. So what this work does, it highlights that there is this fundamental trade-off between beta cell biology, between this function and expansion. And sort of at its core, this study shows that beta cell may have to choose between sort of growing, between proliferating and function. And Tomosyn helps govern that choice, at least. And I think as we study more and more about what, maybe more regulators will come through.
Very well put. Well, Sushant, we want to thank you again for joining us today to share your expertise and to discuss this paper on the role of Tomosyn-2 in beta cell function and proliferation. I encourage the listeners to read the full article, which is in the May issue of Diabetes.
Thank you, Kurt. Thank you, Darleen.
Next up, we'll be discussing an article from the May issue titled Normalization of Insulin Resistance Rather than Hyperglycemia Before Stroke Improves Functional Outcome in a Mouse Model of Type 2 Diabetes. We are joined by co-corresponding author Dr. Vladimer Darsalia, PhD, a docent in neuroscience, which is the Swedish equivalent to an associate professor, and he is in the Department of Clinical Science and Education at the Karolinska Institute. Welcome, Vladimer, and thank you for sharing your work with us today.
Thank you for having me. Appreciate it.
So, Vladimer, before we dig into your work, let's start with an icebreaker. And I guess the question I'd like to ask is, can you tell us something about your personal life and how it might intersect with or inform your work?
Oh, well, one thing actually that was quite touching for me that put me on this path was when I was doing my master's program in Lund in Sweden. I guess that person sent out this email targeting everybody in the research center. I'm assuming he was a recent sufferer of stroke who had been dealing with really bad disability after. And being an athlete in her younger years, it was really weighing heavy on her. Yeah. So this letter, in a way, kind of moved me to try to do my best to contribute in one small way to try to mitigate the disabilities that come after stroke.
I think that counts.
This cemented my path to research line that I pursue now over 20 years.
Yeah,
that's really interesting. To move towards the science, are there any other authors or collaborators that you would like to acknowledge before we talk about the science?
Yeah, certainly. Great thanks to the lead author, Dr. Ellen Welkerstern, who developed the treatment paradigms and dosing regimens that were key to the success of this study. And she achieved a very tight metabolic regulation. It was so important for the project, and her tireless and meticulous work generated all the data. Another person that I should thank, without whom this wouldn't have been possible, is our long-term collaborator from Boehringer Ingelheim, who provide the long-acting glucagon agonist, and also for his insights into metabolic regulations that we neuroscientists were not really that familiar with. And finally, I would acknowledge Dr. Cesare Patrone, who is the co-corresponding author on this paper. And he's been driving the larger research program at our institute, which is dedicated to mostly repositioning diabetes medications to improve stroke sequelae by targeting both metabolic aspects and brain-related complications.
Cool. Why don't you give us an overview? It's just sort of, you mentioned that stroke has been a theme throughout for your work, but why don't you mention an overview of your research program and then maybe touch on how this program came out of that?
Well, to be brief, it might take a lot of time to explain everything, but just to give a kind of an elevator pitch of our research program is, so we study mainly the consequences of diabetes on brain health. And particular emphasis is on stroke, although we also deal with cognitive issues and movement disorders. And we want to mitigate the stroke sequelae by either pharmacological interventions, that's pre-stroke or even after stroke, and also with lifestyle interventions that can affect the metabolism. And the goal is to try to increase brain's resilience to injury and when the injury happens, to promote neural repair and recovery after stroke.
Very cool.
So Vladimir, let's get to this manuscript now, which focuses on stroke outcomes in a rodent model of type 2 diabetes. And specifically, it seems that you've set out to disentangle the contributions of pre-stroke hyperglycemia and insulin resistance. Is that correct?
Correct.
Right. And can you tell us a little bit about the model that you've established to segregate those conditions and the rationale behind it?
Sure. I'll try to be brief on that. It's a great question because this is normally how we start the conversation, where we try to explain people what is the connection between stroke and diabetes. And just to go back a little bit in the background, now it is a common knowledge that people with diabetes, they experience worse outcomes after stroke, right? And among them, those who manage their diabetes more effectively, they are generally better off with milder neurological sequelae. There were a few observational studies that also showed that both insulin resistance and hyperglycemia, they independently associate with worse stroke outcomes. What was interesting that reduced insulin sensitivity was also a major contributor to worsened outcomes in non-diabetic stroke patients, right? So in total absence of hyperglycemia, insulin resistance was still making stroke outcomes worse. And then to connect this, we need to look at the very important function of insulin signaling in the brain. Insulin receptor, which is widely expressed in the brain on both neurons and support cells, and is doing many other functions above glucose regulation that you are normally used to hearing about, right? In healthy brain, insulin signaling helps neurons to maintain connectivity. It facilitates learning, memory. It also helps the brain to manage energy. It's acting on the astrocytes, which are the helper cells, which coordinate fuel supply and metabolic support to neuronal circuits. And interestingly, after injury, like stroke or TBI, traumatic brain injury, these broken neural connections must be reestablished to achieve functional recovery, right? And in this context, insulin signaling is protective. It's linked to beta cell survival, neuronal circuit restoration, and reduced inflammation. So, already before we started our studies, there was plenty of data pointing that dysfunctional insulin signaling was the major driver of worse stroke outcomes in people with diabetes. Now, going back to experimentally, the first hint that we saw that the insulin resistance was important for stroke recovery, I think it was like 5 years ago, when we compared short versus long-term weight loss in obese mice, and we looked at their effects on stroke outcomes. Just very briefly, so what we did, we replaced obesogenic diet with the standard maintenance chow, right? For 2 or 4 months before mice were subjected to stroke. What we observed was that despite both groups achieving similar weight loss and fasting glucose, the 4-month group was doing much better in terms of motor recovery. And the only meaningful metabolic difference we saw between these two groups was much better insulin sensitivity in 4-month weight loss group. And this prompted us to start thinking about specific experiments that could help to individually understand the contribution of both hyperglycemia and insulin resistance to stroke outcome. Now, at the time, of course, there were no tools available, and we attempted to do so in, I think, 3 years ago in 22, when we induced insulin resistance without hyperglycemia or obesity in mice. We saw similar worsening of motor recovery. And this reinforced the insulin resistance hypothesis by showing its importance. But since the model was neither obese nor hyperglycemic, we could not really address the question of hyperglycemia contribution. In the end, it came down to the availability of pharmacological tools. So as you know, any intervention that increases insulin sensitivity would eventually reduce blood glucose. So the disentanglement of IR and hyperglycemia was almost impossible at the time. And then luckily for us, we have this eureka moment. It's a funny story. I believe sometime 2 years ago. So we were discussing different weight loss strategies with Dr. Klein that I mentioned before. And he mentioned that they were experimenting with long-acting glucagon agonists to promote weight loss, right? And the results were really spectacular in that regard. But unfortunately, he thought this compound had no clinical future since it increased blood glucose, right? So for him, it was something that wasn't going anywhere. And then it was like everything fell into place for us. So we could induce weight loss with glucagon. As you know, weight loss is the best way to reduce insulin resistance. And then at the same time, keep blood glucose high, as long as there was a drug around. For the comparison, what is the opposite of glucagon? It's insulin, right? So it's a very good glycemic regulation with insulin, with zero improvement of insulin sensitivity. And on the other hand, weight loss with glucagon with very nice improvement of insulin sensitivity, but maintaining the hyperglycemia. So once we got these tools in place, then the rest was rather simple.
And so you made a deliberate choice to stop the treatments about 72 hours before inducing the stroke. Can you tell us a little bit about the rationale for that, and then maybe get into a little bit into some of the results?
Yes, yeah, great question. So what we wanted to achieve here was to isolate metabolic effects on stroke recovery. So without risking contamination with direct neuroprotective action, right? Insulin is well known to protect the brain from injury. So we definitely didn't want it to be around while animals were experiencing stroke. And there is also some evidence, also less robust, for glucagon acting in some neuroprotective way. It hasn't been confirmed with many studies, but we decided to avoid any risks. So that's why we had this washout period to try to get rid of the circulating compounds.
Yeah. And then you focused on motor control and saw some effects there. Did you look at any cognitive endpoints? You mentioned that you were interested in that as well.
Yes, great question. The only problem with that is our stroke model. We use transient occlusion of middle cerebral artery for 30 to 40 minutes, and this induces damage in the striatum. So it's the brain area that is main input hub of basal ganglia, and it integrates signals from cortex and thalamus and helps control voluntary movement and shapes what actions get expressed. So additionally, in the same area, we have an internal capsule. It's a dense white matter highway that carries major descending motor and ascending sensory fibers between the cerebral cortex and the brainstem, spinal cord. And it goes right through it. So in effect, our stroke model localizes injury primarily to sensory and motor circuits, right? So that's why we chose to go directly for the motor tests. Another thing that needs to be considered here is that probably there are some cognitive components because striatum is in a way involved in some way with cognition, but it is difficult to isolate from the motor components because many cognitive tests that we were looking at least in mice, they rely on free movement or choice of direction. And sensory motor impairments interfere with that.
Yeah.
So we are trying to develop these methods to let's say, try cognitive tests once the motor symptoms have gone away, but we don't know if we're going to be successful or not.
So, Vladimir, I think that takes us nicely to another question I have, which was your finding that the infarct size, right, was similar between groups, but their outcomes were so dramatically different. Can you maybe give us some insight into that?
Yes, of course. I mean, we actually aimed for this, and then we got quite lucky to get these results, right? So let's go a bit deeper. One of the reasons we chose to implement the off-treatment period that you mentioned before was precisely to avoid any differences in infarct size. As I said, we got lucky that metabolic effects alone were not enough to create such a difference. Because if that were the case, we would have ended up with an interesting but rather boring results, if I have to think about it. Smaller infarct leads to better motor outcomes. Nothing to see here, right?
Yeah.
Right.
Instead, with the similar infarct size, it told us that the better recovery after insulin resistance normalization is driven by improved brain's self-repair capacity or neuroplastic capacity, which nicely ties into the role of brain insulin signaling in neurorepair. And this is something that we intend to investigate further in the future.
Yeah. Yeah. So that brings us to the next question about the systemic inflammation and the neuroinflammation. And so, how do you think the pre-stroke insulin resistance kind of primes the brain to kind of have that worsened inflammatory response?
Great question. So when body is insulin resistant, it in a way sets up the brain with a kind of a low-grade pre-inflamed state. Right? This happens in several ways that we know of. So the one is that insulin resistance weakens insulin's normal protective signaling, and it biases the glial or vascular cells towards stronger innate immune activation after the injury. It also makes the blood-brain barrier more fragile. So when stroke releases like danger signals, it's easier for inflammatory cues and the immune cells to escalate the response. After stroke, the same ischemic insult triggers a bigger inflammatory cascade when you are insulin resistant, because more cytokines, chemokines, more recruitment, more secondary damage, because the system is already primed.
Yeah.
And this is where the MIP1B ties in, because this signaling directly promotes trafficking of leukocytes and even disrupts the barrier function. So keeping it low in the better recovered group is consistent with a less amplified post-stroke inflammatory loop, so to say.
So I want to maybe look forward a little bit now. It seems that there's some direct implications for how we might treat type 2 diabetics, both prior to and maybe after strokes based on your work. Can you maybe give us some key takeaways from this paper and then where you're going forward with that?
The key takeaway for us would be that insulin resistance seems to be the main roadblock to functional recovery after stroke, at least in type 2 diabetes. And it works likely by limiting the brain's repair capacity, and it amplifies inflammatory responses. The fact that improved recovery after insulin resistance normalization was achieved without infarct size change supports this hypothesis. For future questions, I think we will devote next 2 years to understanding of the mechanistic aspects of insulin resistance, or specifically the brain insulin resistance, how it affects the brain health, and what are the molecular pathways, perhaps downstream insulin signaling, that can become their own targets to improve stroke outcome, more specifically than just broad targeting of insulin signaling.
That's very cool. It feels like it has a lot of implications broader than just stroke, right? Maybe Alzheimer's, dementia, et cetera.
Yeah, exactly. When it comes to insulin specifically, the people have been trying to stimulate insulin signaling to halt the progression of Alzheimer's, to try to initiate neuroplasticity. So anything that involves brain plasticity, even, for example, myosensing prosthetics that need brain remapping to adjust, right? It might benefit from facilitating insulin signaling and promoting neuroplasticity.
Yeah. Vladimir, thank you so much for joining us today and sharing your expertise and discussing your recent work on the roles of pre-stroke hyperglycemia versus insulin resistance on post-stroke outcomes. I encourage the listeners to read the full article in the May issue of Diabetes.
Thank you for having me.
Welcome to another episode of Sweet Talk. I'm Kevin Williams, and I'm joined by my co-host, Dr. Darleen Sandoval and Dr. Kirk Habegger. Today, we have Dr. Timo Mueller, who is director of the Institute for Diabetes and Obesity at Helmholtz in Munich. He's also professor at the Walter Straub Institute for Pharmacology and Toxicology at the Ludwig Maximilian University in Munich. Timo has played a central role in advancing the science behind modern obesity and diabetes therapeutics. He is also the current recipient of the American Diabetes Association's Outstanding Scientific Achievement Award. Timo, congratulations on this well-deserved recognition from the ADA, and welcome to Sweet Talk.
Thank you very much, Kevin, and thanks a lot for having me.
Now, we start these things off with a little bit of an icebreaker, and we were curious, you've had all this success in the laboratory, but outside of the lab, are there any hobbies or interests you might you might have that would surprise anybody, maybe something that helps you reset and recharge?
Yeah, apart from my family, I'm a very passionate scuba diver. I love to spend time underwater, very quiet, and watch the fish. And when you see something big crossing you, it really shows you that your adrenals are still working. So this is pretty exciting.
That's really
nice.
cool, Timo. I didn't know that.
Yeah.
Now, around Munich, you probably don't have many diving opportunities. Is there a favorite spot?
Yeah, that's actually the downside, right? So you need to, I also like to go scuba diving in warm water, right? So I typically used to go to Egypt 2 or 3 times a year, but I also used to go to the Maldives once a year because they have pretty big sharks there. They also have very nice tiger shark areas where you can basically scuba dive with tiger sharks, and it's very exciting.
Wow. That's really cool.
Now, I have to say, when Timo and I were postdocs in the same lab, that I hated his scuba diving because that meant I had to take care of all of his mouse studies.
Which at the end worked pretty well, I have to say. So I'm very grateful that you did this, Kirk.
Well, no, I'm obviously just kidding, Timo.
Yeah,
Kirk,
you
are
know,
you gonna
Kirk
get an
is
acknowledgement
just
slide?
exactly. Kirk is just
That's right.
vying for some
You gotta get-
acknowledgments during the presentation.
You
Yeah.
have to get those article citations however you can.
All right, Timo, before we dig into your science, tell us about young Timo's journey and how did you get into obesity and diabetes? What got you interested? Were there particular questions that that's really struck you? How did it all start?
Yeah, that is actually a very interesting question. So I remember when I left kindergarten, we received a letter, and I found this letter, I don't know, 20 years later in our cellar, saying that Timo wants to become a biologist. Right? So for me, it was clear from the beginning that I wanted to do something with biology. I have to say that I had no idea what biology is when I left kindergarten, but I studied biology, animal physiology and zoology, and I always wanted to become a marine biologist. The problem is that the place where I studied, there was no sea, so I couldn't do marine biology. So I wanted to do physiology. And I had a professor that was very close to me, and I wanted to do my master's thesis in his lab. And he was a physiologist, and he told me, don't come to me because you will not find a good job. So you need to do obesity and diabetes research, which is downstairs. So he sent me downstairs to the lab of Martin Klingspor and Gerald Heldmeier, who were doing studies on diabetes and obesity. And this was basically my start of my obesity career. And then I did my master's thesis in the lab of Martin Klingspor and Johannes Hebebrand. And then they basically paved the way to my PhD thesis, which I did in the middle of Germany in Essen, right? In the lab of Johannes in the lab of Johannes Hebebrand. And Johannes was doing genetic studies on human obesity. And Johannes was also the one that in my PhD thesis sent me to Cincinnati to Matthias Chöp to do a little mouse study. And this was then the time where I met Matthias Chöp in Cincinnati. I spent around 3 months in his lab. This was also the time where I met you first, Darleen. And this was before I joined Matthias as a postdoc. And Matthias immediately offered me a postdoc position. And then a couple of years later, 1.5 years later, I met Matthias in Germany on a conference. And this was right 2 weeks before I was about to submit my PhD thesis. And he immediately reminded me that I should come to Cincinnati to work in his lab as a postdoc. And then actually a couple of weeks later, I was flying to Cincinnati and started my postdoc in Matthias' lab. And then in the first week, I met Richard DiMarchi in Matthias' lab and was introduced into the universe of polypharmacology. I was very skeptical at the beginning, but I was convinced very, very rapidly that this is really a fruitful way of pharmacology and how we can potentially develop drugs to treat obesity and diabetes. And then I was at this point the only postdoc in Matthias' lab. And then a few months later, Kirk Habegger joined the lab, and I had a great time with Kirk. And at the beginning, I had a lot of joint projects with Kirk. Right?
That's true. That's very true. So
Really
Timo,
cool.
your comments on polypharmacy actually take us to our next topic, which is you've done a lot of work in the incretin axis and agonization of GLP-1 receptor and then GIP receptor. And GLP-1 has always been more intuitive to people, I think. Easier to wrap their heads around why this would be a good anti-diabetes, anti-obesity therapeutic, whereas GIP is always a little more complicated. When you explain the rationale for GIP, how do you usually frame this?
Yeah, so there are several reasons why people look at GIP with a lot of skepticism, right? So first of all, mice that are deficient of the GIP receptor are resistant to diet-induced obesity, which naturally implies that you should inhibit the receptor rather than activating it to treat obesity, right? But the other thing is that also humans with type 2 diabetes are kind of resistant to the insulinotropic effect of GIP, which accelerates the skepticism against GIP. The other thing is that GIP by itself, the native GIP, has very little effects on metabolism. You need to have it in a long-acting form with sufficient exposure in the brain in order to really peel out the benefits of GIP in the brain, right? It's a little bit similar to the GLP-1 receptor agonists, right? When you compare liraglutide and semaglutide, the structure is almost identical, but it's the half-life and the exposure you get in the brain that really makes a difference. And this is also the case for GIP. Another reason why GIP is regarded with a lot of skepticism is because GIP is a bifunctional hormone, right? It can not only regulate blood glucose by either stimulating insulin secretion or stimulating glucagon secretion, depending on the level of blood glucose. It can also either stimulate lipolysis or induce lipogenesis, depending on the energetic status and the state of insulin resistance, right? When you are insulin sensitive, GIP will help the lipids to find their way into the adipocytes where they belong to. But when you are insulin resistant, GIP will stimulate body weight loss and lipolysis, right?
Right. Thanks.
And you touched on it a little bit there of the action of these incretin-based compounds to act in the brain. And I was just curious, your work has helped to establish the action of GIP and these long-acting agonists to act in the brain and the biology behind it. But what do you think might be the most important conceptual advance there of what it does to help explain how these peripheral models might not have been able to explain?
Yeah, so the key important thing when we talk about GIP is that we need to peel about the physiological from the pharmacological effects. This is way more important relative to GLP-1 because GLP-1, if you dose it correctly and highly enough, it will also decrease body weight. But for GIP, it's really important that you have it long-acting. And the reason why we became interested in GIP receptor agonism relative to antagonism was a study that we did still in Cincinnati with Nikki Parker, where we used a long-acting GIP and gave it in a physical co-mixture with GLP-1. And all of a sudden, we saw that GIP agonism accelerates GLP-1-induced weight loss, which was pretty unexpected. We thought that it would accelerate the insulinotropic effect of GLP-1, but that it accelerates also GLP-1-induced weight loss was pretty surprising for us. And then we repeated the study a couple of times, and the result was consistent. And this then actually prompted us to basically fuse the sequence of GLP-1 and GIP into a single molecule. And what is also quite kind of interesting, GIP accelerated food intake inhibition of GLP-1. And based on my understanding, everything that decreases food intake has to go over the brain. We didn't understand the mechanism, but the fact that the drug accelerated food intake inhibition clearly pointed to a role in the brain. And this was then after we moved with the lab to Munich, we started to generate mice that lacked the GIP receptor in the central nervous system. And then we observed that mice that lacked the GIP receptor in the CNS are pretty much resistant to the body weight lowering effect of GIP. And this was then further supported by studies from Cambridge, where they used a dread in order to activate the GIP receptor containing neurons. And when they activate these neurons in the hypothalamus, they see that the mice start to eat less, indicating that the GIP receptor is sitting on the right neurons. And this was then confirmed in our knockout studies. And As a next step, we then asked the question, which neurons are responsible for this effect? And then we figured out that the GIP receptor is highly co-localized with inhibitory GABAergic neurons with a marker called VGUT. And then we knocked it out in the VGUT positive neurons. And then we observed pretty much the same phenotype as when we eliminate the GIP receptor in the entire CNS, indicating that GIP acts via these GABAergic neurons to inhibit food intake and to decrease body weight.
You know, I think that's a really cool aspect of the story, and that GIP acting through GABAergic inhibitory neurons, whereas GLP-1 has been implicated to be acting primarily through excitatory neurons, and that you have these two incretin molecules in the periphery, but in this case, these are long-acting versions of those factors that are able to somehow in design signal through the two major neurotransmitters are either driving excitation or inhibition, and that is what appears to be important in enhancing either one of the effects of those incretin-based compounds on food intake and body weight. I just think that's a really interesting aspect of the biology there.
Yeah, absolutely right. So you have to keep in mind that when you take a long-acting GIP and you put it into a GLP-1 receptor knockout mouse, it will decrease body weight equally to a wild-type mouse, right? So the GLP-1 receptor pathway is not important, not relevant for the weight loss that you see by GIP receptor agonism. So it's completely separate pathways, which makes it also attractive in terms of pharmacology, because you are hitting two pathways which, independent from each other, decrease body weight, right? Which is exactly what you want in polypharmacology.
Timo, just as a quick follow-up, you talked specifically about the hypothalamus and GIP receptors there. We know that they may be in other important nuclei. Are you exploring those as well?
That's true. So the hypothalamus actually does not seem to play such a great role for the body weight lowering effect of GIP. It's more GLP-1 that goes via the hypothalamus. The GIP seems to like the area postrema of the hindbrain better in terms of decreasing body weight, right? Whereas the GLP-1 pathway needs the hypothalamus. Yeah.
Right. Thanks for that clarification.
So let's go back to the beginning. You said that you were skeptical of the polypharmaceutical strategy. So what was it that made you skeptical? And was it that study with Nikki that sort of turned it around, or were there other points of evidence that started to direct you towards this idea?
Yeah, absolutely. You have to keep in mind that I came only with my textbook knowledge in biology into Matthias' lab, right? And Matthias and Richard are famous for translational broad hypothesis, right? And when I came into the first lab meeting and I heard that, and this was the time where we talked about a GLP-1 glucagon coagonist, right? And my textbook knowledge was GLP-1 decreases blood glucose and glucagon is doing the opposite, right? So my skepticism was, are we not neutralizing the glycemic effects here, with the result that we don't see any effect? And this was actually my skepticism. But after I saw the data, and the data are really convincing, right? I quickly understood that there is so much more flavor into this concept as I could imagine at the time. And I was convinced very, very rapidly, because the studies were highly reproducible, right? And glucagon can even improve glucose metabolism via its ability to decrease body weight. It even improves glucose tolerance, more powerful than Extendin 4, right? It's very, very powerful molecule to improve glucose metabolism.
So let's talk about GIP and what it adds. And specifically, it's really fascinating that both agonizing and antagonizing the GIP receptor pretty much get you to the same place, right? So how do we make sense of all this? And what does it tell us about the mechanisms that really are driving these effects?
Yeah, this is really an interesting thought, right? So you're absolutely correct. GIP receptor agonism and antagonism both accelerate GLP-1 induced weight loss. And based on our data, and I know that other people can also confirm this, and we will very soon see another big paper on that topic, is that it looks like GIP receptor agonism and antagonism decrease or accelerate GLP-1 induced weight loss via different mechanisms, right? So the first thing that we note is that the agonist needs the GABAergic GIP receptor neurons in order to decrease food intake and body weight. But the antagonist does not need these GABAergic neurons. So the animals that lack the GIP receptor in the GABAergic neurons are completely susceptible for the ability of the antagonist to accelerate GLP-1 induced weight loss, whereas the agonist has no effect in these mice anymore. So this indicates that we are talking about different mechanisms. In our hands, it looks like the antagonist is somehow inhibiting the GLP-1 receptor neurons in order to be fully efficacious. And when you inhibit the GIP receptor on these neurons, they lift the brake from the GLP-1 receptor neurons, and the GLP-1 receptor neurons get more susceptible, more efficacious after treatment with GLP-1. In other words, the antagonist makes a better GLP-1. The GIP receptor agonist, and we just talked about this, does not need the GLP-1 receptor in order to decrease body weight. So we are talking about different mechanisms of how agonism and antagonism affect GLP-1 induced weight loss. The agonist is independent of GLP-1, and the antagonist accelerates GLP-1 induced weight loss. And we have basically provided first data on this in a paper that we published last year, and there will soon come another paper from another group that will show that this is also separated in different areas of the brain, which is pretty exciting.
Right.
So, Timo, as a follow-up to that, these drugs they're one drug, right? They're not breaking apart. So they're biased towards one receptor or another, correct? And so is it just that they're able to hit a specific population sufficiently to have these opposing effects?
So they are one entity, these drugs, right? They are one entity with either balanced activity at both receptors or unbalanced activity. Tirzepatide, for example, favors the GIP receptor 5-to-1 over the GLP-1 receptor, but most dual agonists that we produced together with Richard are kind of balanced. So they hit both receptors with equal potency, right? But depending a little bit, you can play with each molecule has its own balance, right?
Yeah.
So it's either 1-to-1 or unbalanced like tirzepatide. Yeah. But
And
do you
know what the Amgen drug, what its balance is? Any idea?
Well, I mean, the Amgen drug neutralizes the GIP receptor and activates the GLP-1 receptor. So I'm not sure whether we can talk about the balance because it will silence the GIP receptor, right? But the Amgen drug is an antibody linked to two GLP-1 receptor agonists.
All right. So what's next for the field in terms of therapeutics? And what problems do you think there are either that your lab is really interested in solving or that the field needs to address for treatment obesity to optimize treatment?
Yeah. So to first answer the question, what I think is next. So we have a paper in press at Nature where we developed a quintuple agonist. So one molecule that activates 5 receptors at the same time. But this one is a kind of smart molecule because we are sending a triple agonist PPAR into the incretin receptor positive cells. And we have then activation of all 3 PPAR receptors plus GLP-1 plus GIP, right? But only in the incretin receptor positive cells. And this molecule decreases body weight up to 50% with up to 80% loss of fat mass and an unbelievable improvement in insulin sensitivity. Yeah, so this is, I think, one of the strategies that I think will be really fruitful, that we can use the incretins as a carrier to carry nuclear acting hormones specifically into the incretin receptor positive cells. Since the PPARs are not really famous for decreasing body weight, it's actually rather the opposite. They rather increase body weight. How they work is the PPAR goes into the incretin receptor positive cells and decreases inflammation in the obese incretin receptor positive neuron, with the consequence that the incretins work more efficiently. Right? So this is, I think, the way how these molecules work. The paper will soon be out. I'm pretty sure before the ADA meeting. And related to your second question, the unmet clinical needs, I think it's not so much the weight loss efficacy, right? Because most people on these medications do not reach the highest approved dose because they are happy with a lower dose and the weight loss they experience with a lower dose. It's more the side effects, right? So to avoid the typical GLP-1 induced side effects is certainly something that is really one of the high priorities of next generation therapeutics. And we just published a manuscript together with Blue Water Bioscience, a company that I co-founded with Richard DiMarchi and Matthias Chip. And we have one molecule in the pipeline that has tirzepatide, retatrutide-like weight loss, but without GLP-1, right? So we think that this molecule can basically have a better tolerability profile relative to the GLP-1 containing drugs. So this is certainly one thing. And the other thing is, of course, that we can now make more patient-oriented, personalized therapeutics that are specially tailored to the individual patient needs. For example, next to decrease body weight, we can also emphasize more to treat atherosclerosis, fatty liver disease, and so on, right? So maybe even preserve lean mass or increase lean mass in the elderly, which is also a good thing, right? So I think that we, with this polypharmacological concept, opened a toolbox that we can now use to generate a whole variety of drugs that go from lower side effects to more effects in the liver or more cardiovascular benefits. So I think we are living in very excited times. We have to keep in mind that we tried now for more than 100 years to pharmacologically help people to decrease body weight with overall very limited success. And since 15, 16 years, all of a sudden managed to come up with strategies that really so far turn out with a favorable safety profile. So it's very, very exciting.
So Timo, do you foresee that we are leaving GLP-1R pharmacology, or do you see it as a sequencing of therapies as a patient progresses from diseased to a healthy state?
Well, I do not want to downplay the therapeutic value of GLP-1 because GLP-1, for those who tolerate the GLP-1 therapeutics, it's a wonderful drug, right? So GLP-1 is also the more powerful weight loss drug relative to GIP, right? So there's nothing wrong with GLP-1 as long as you tolerate it, right? The problem is when you don't, right? So if you experience GI distress, nausea, vomiting, right, you want to have another drug. And having an alternative to these GLP-1 therapeutics is valuable. Maybe not in the same range of weight loss relative to GLP-1, but certainly in a range where the patient has metabolic benefits without the GI side effects that these people unfortunately need to experience when they take the GLP-1 therapeutics. Yeah.
I was also curious, you talked about the magnitude of weight loss and how it is going up with these unimolecular polypharmacology as the targets are increasing. And you talked about a compound that you have in press now of up to 5 targets, I think is what you mentioned. And the percentage of weight loss is going up. I think probably, correct me if I'm wrong, but the paper that you probably have in press is a preclinical assessment. I'm curious, in the clinic, as these more targets are being hit, is it increasing efficacy not only in magnitude, but the percent of patients that are also losing weight? Because there are a specific group of individuals that have a resistance. There was a recent study that came out that suggested that there could be some mutations in GLP-1 receptor itself that might be indicative of why these patients might be resistant, and these other targets that could be in the polypharmacology and these unimolecular drugs might be able to assist those individuals. So I was curious if you had any insight into that.
Absolutely. So you are 100% right? So there are certain mutations that we know that affect the efficacy of the GLP-1 based therapeutics. So the more independent pathways you are activating, the advantage is that it's less likely that the patient will be 100% resistant to all of these different pathways, right? The other benefit you have is that when you want to have a maximal body weight loss with a single drug, you need pretty high doses. That's the reason why liraglutide or Victoza and Saxenda differ by a factor of 2 in the dose, right? The same for semaglutide, right? So Wegovy and Ozempic differ in a factor 2 in the dose, depending on whether you want to treat obesity or type 2 diabetes. The brain generally needs a higher dose of the drug. If you are activating now multiple targets, this allows you to go down with the dose at each receptor, which at the end should be also beneficial for the tolerability of the patient, right? So just staying with the quintuple agonist, right? So we used lanifibranor, which is a pan-PPAR, and we used 10 nanomole per kilogram in our studies, which is roughly 8,000 times lower in the dose relative to what you need when you take lanifibranor preclinically to have metabolic effects on the liver, right? So the targeted design and the polypharmacology allows you, by targeting multiple pathways, to be sub-threshold with the dose to have metabolic impairments, right? So side effects, right? So this is a very important note here, right?
One final question we have is related to this award from the American Diabetes Association, a well-deserved honor, and it does recognize independence of thought, originality. In your career, was there a point where you had to push against consensus to pursue something that you believed in? And if you were to tell a young scientist today facing that same choice, what would you give them advice on?
Yeah. So first of all, thank you very much. This is certainly the greatest award that I received so far. So I'm I'm very happy and very grateful for seeing our work being recognized by such a prestigious award. But related to your question, you are absolutely right? So when I started my career in the lab of Matthias Zschöp, right? So we started to convince the field not only that GIP is doing any good, but also that activating the glucagon pathways in order to treat diabetes was something that intuitively is pretty difficult to deliver to the field, especially to clinicians who are seeing patients on a daily basis, right? So this took quite some time, and it needed at the end the clinical data to really prove our preclinical concepts valuable. Right, so swimming against the current is something that I learned since many years. Now we are also building a bridge to scuba diving again, right? But yes. So in pharmacology, a lot in pharmacology is related to dose and co-therapy, right? So just staying a little bit more in the GIP field, right? So giving GIP to humans with type 2 diabetes will likely result in a diminished insulinotropic effect. But if you give the GIP together with a substance that is able to overcome the hyperglycemia, then all of a sudden the GIP will start to work again, right? So pharmacology has always different flavors. It's not just black and white. We see the same with the GIP receptor antagonist, right? So it's not so much trying to explain how GIP and agonism and antagonism decrease body weight via the same pathway. We have to accept that it's likely via independent pathways, right? So in Germany, we say there many roads go to Rome, right? So, and the same is the case for obesity. It's a heterogeneous disease, and the brain regulates food intake by different mechanisms. And it is very likely that GIP receptor agonism and antagonism are perfectly doing the job, but they're doing the job via different mechanisms, right? So going against the current and following hypothesis is a lot of fun. I mean, this is really the best day, the best hours of my day is when people come to my lab and show me data that totally go against current way of thinking, because this is really what makes my life so enjoyable, right? Thinking about new ways of explaining data.
That's great. And I might make a suggestion. In Munich, you could say many roads lead to Oktoberfest or the beer garden or something.
That's very true too.
Timo, this has been a great conversation. Before we let you go, though, where can the listeners follow you and your work? Do you have a lab page, an institutional page, anything you could point them to?
Yes, so I have an account on Instagram, and I'm also on Facebook, and I have a LinkedIn account, and I have a Twitter, an X account. Yeah.
Timo, you'll have to check back in with us or come back on the program and let us know how many followers you picked up from the advertisement. But thanks again for joining us. And to our listeners, as always, please like, follow, and subscribe for more Diabetes Bio content.
Thanks to all of you for joining us on the Diabetes Bio podcast. We hope that you're enjoying our discussion of the work that the Diabetes Journal is publishing each month. The feature articles we discuss in each episode are freely accessible at diabetesjournal.org/diabetes and are also listed in the episode notes for your reference. As with any podcast, it really helps us if you like, rate, follow, and subscribe to the podcast. And if you have any feedback for us, we'd love to hear that as well. We look forward to presenting to you each month.